Hello, my name is Emily DeVoe, and I will be talking about the synthesis and functionalization of cerium oxide nanoparticles. So I'll start off with the summary. The reason we're looking at cerium oxide nanoparticles is there are many medical applications. They have antioxidant, anti-inflammatory, and bacteriostatic properties. Cerium oxide nanoparticles can also be used in tandem with other treatments such as photothermal therapy to give a more focused treatment to prevent cell death of healthy cells, which is something that is very common in cancer treatments. And what I will be showing today are two methods of synthesis as well as functionalization of those nanoparticles. So the introduction. The applications of nanoparticles include involving in supercapacitors for increased efficiency, antibody functionality for targeted delivery. It causes apoptosis of cancer cells, and it also can be evolved in tissue engineering for biomedical applications. And most importantly, the biocompatibility. Nanoparticles are biocompatible, meaning they can freely go throughout your body without causing much damage. Now, they have the following health benefits. They are antibacterial. They're antioxidants, meaning that they reduce the free radicals within your body. They have anti-cancer properties. They can be used for targeted drug or gene delivery. And new studies are showing that they have anti-diabetic properties. So I will be looking at the antibacterial activity right now. So here's the first schematic of the antibacterial activity. There are two different types of contact in which the nanoceria can enter the cell. Firstly, direct contact has the cerium oxide nanoparticles enter the cell via endocytosis, where once inside the cell, they react with the ribosomes, the mesosomes, and the nucleus to cause the DNA to be denatured and causing cell death. This is a bacterial cell, so this is a good thing. And then indirect contact is where the nanoparticles don't actually enter into the cell. Rather, they are attracted to the cell wall where the reactive oxygen species are released outside of the bacterial cell wall, and then once inside the cell, it completes the same thing as the direct contact where the reactive oxygen species denatures the DNA and kills the cell. A main cause of study is free radicals, and some of the sources of free radicals are UV rays, atmospheric pollution, smoking, of course. Stress and poor diet are also some things that can contribute to other free radicals within the body. So firstly, free radicals, they're not very good. They cause very high oxidative stress in the cell, causing denaturation, disruption of cellular components, and it just leads to cell death because the cell cannot handle it. However, once the oxidative stress has begun in the cell and a cerium oxide nanoparticle is introduced, the nanoparticle is able to scavenge for free radicals, returning it to a healthy cell. The cerium oxide +3 is oxidized and with a radical to produce H2O2. And this is then reacted to give reduced cerium and oxygen. So it takes out all of the damaging things in the cell and returns it to physiological, healthy condition. Now, if we look down here, we can see another graph. Firstly, I'll look at this side. This is the normal cell. So you've got the cerium oxide nanoparticles and the targeting agent. And this happens through endocytosis, where the nanoparticles enter the cell. It reacts with the endosome inside the cell, forming the complex to release the nanoparticles. These nanoparticles then scavenge the reactive oxygen species within the cell to keep the cell healthy. There's superoxide dismutase activity that happens within the cell, and because of the physiological pH, there's also a catalase reaction that turns the H2O2 from the SOD activity into water to make it so that it's a healthy, normal cell. However, over here in the cancer cell, it has overexpressed folic acid receptors. So the nanoparticle is mediated through receptor-mediated endocytosis entering the cell. The superoxidant dismutase mimetic activity takes the reactive oxygen species to produce H2O2 as over here. However, the low pH inside of the cancer cell prevents the catalytic activity, producing more reactive oxygen species. This causes mitochondrial disruption, protein oxidation, damage to the nucleus, and eventually DNA denaturation, leading to cell death. And this next one is talking about targeted delivery. We have the cerium oxide nanoparticle, which is functionalized with the drug. Then once we have the drug-loaded cerium oxide nanoparticle, we can functionalize it with a targeting agent. This complex then enters the cell through receptor-mediated endocytosis. Now inside the cell, there is a high level of glutathione and a low pH. The glutathione forms a complex with the cerium oxide nanoparticles, releasing the drug, and the low pH prevents the catalytic activity, which prevents the H2O2 from turning into water, meaning that more reactive oxygen species are produced and the cell is damaged, causing cell death. Okay, so here is the next graph. We've got the cerium oxide nanoparticles, and here we have doxorubicin, which is an anti-cancer drug. Now this drug is highly cardiotoxic, meaning that it is very important for targeted delivery. So once we have the cerium oxide nanoparticle, which is negatively charged, we have the doxorubicin attached to it since it is positively charged, giving an electrostatic interaction. This goes through the cell through endocytosis, and the high levels of GSH cause the drug to be released. Causing endocytosis of the cancer cell. Now, the second functionalization done to the nanoparticles for this project is dextrin functionalized. Now, the reason for functionalizing with dextrin involves changing the shape, size of the nanoparticles. It's more biocompatible, and the complex with the glutathione in the cancer cell releases the cell drug. So what is happening here is some dextrin, and it is put onto the cerium nanoparticle via electrostatic interactions. What they did here is they took a cell culture, they left it for 24 hours, and then introduced the particles to the cells for 1, 3, and 5 days, showing a significant decrease of cancer cells survival after the 5 days, which can also be the viability can be tested with an annexin test or a fluorescence test. So now that we know why we're doing what we're doing, I will go ahead and talk about the materials and methods. So the bare nanoparticles were produced using the precipitation method. Ammonium cerium nitrate was reacted with urea along with water for an hour, refluxed at 100 degrees Celsius. If there was precipitate, then the reaction was continued for an additional 4 hours. After then, we got the cerium oxide nanoparticles, which were cooled and washed with deionized water, and the sample was diluted to a 1% weight of solid. The next method was the doxorubicin functionalized nanoparticles. It's put on via electrostatic interactions, enters the cell through endocytosis, and then damages the inside of the cell, as I said before. And it was found that the nanoparticles have a loading ratio of 5 to 1.45, meaning that there is approximately 5 doxorubicins that can be placed onto the nanoparticle. Finally, we've got the dextrin functionalized nanoparticles, and as I said before, dextrin is very important for editing the size and shape as well as the biocompatibility of nanoparticles. The dextrin was added to cerium nitrate and ammonium hydroxide and added dropwise. Then it was stirred for 24 hours at room temperature, centrifuged at 4,000 RPM, and then stored at 4 degrees Celsius. The results are over here. We have a TEM scan of the nanoparticles, and here is the dextrin functionalized nanoparticles. Both of these are within the range of 3 to 5 nanometers, which is very good. Anything classified as nanoscale is anything 100 nanometers or below, so that's definitely fitting that criteria. The doxorubicin functionalized nanoparticles are shown right here. We've got the first line is the doxorubicin functionalized nanoparticles, and the bottom line is the cerium oxide nanoparticles, showing the confirmed addition of doxorubicin onto the nanoparticles. Onto my data, I've got this is for the dextrin functionalized nanoparticles. There's dextrin suspended in water, you've got the water peaks and the dextrin's peaks, and then over here I've got the cerium oxide nanoparticle IR scan showing that there are additional cerium oxide nanoparticles with the dextrin on there. An absorbance scan showed that there is maximum absorbance at a peak of around 403 nanometers, and this value is proportional to the amount or concentration of nanoparticles present. For the bare nanoparticles, here is a picture of how they were produced, the precipitate down at the bottom, and this is an IR scan showing the major peaks of the nanoparticles and confirming the synthesis. So in conclusion, the functionalization and synthesis of cerium oxide nanoparticles is only the beginning when it comes to medical applications, and this technique has much more to offer in research in the subject as it progresses, and then further studies of this project will be on the release of the drugs for cancer. Thank you.